All right, so emptying of the stomach is both under neural, so stretch receptors and such, and the hormones. You got to remember that when you get into that first area of the small intestine, there are enteroendocrine glands, and they secrete hormones and all. And I'll tell you what those are and such. So things that encourage your stomach to move things through is stretching it, putting things in there, partially digested proteins, caffeine, coffee, tea, alcohol, such a little beer, a little wine, maybe a little bourbon. No hand sanitizer, don't do that. And then this hormone gastrin, that'll do it as well. Things that slow it down. When food enters the small intestine, it inhibits the parasympathetic, slows down the secretion, slows down the motility of the stomach. That's the enterogastric reflex. Hormonally, when food enters into the small intestine and such, especially sugars, glucose, and then your fatty acids and all of that, you'll release secretin and cholecystokinin, or as your friends say, CCK. So this will decrease the gastric secretions, and then this one, the cholecystokinin, will inhibit the stomach emptying. Hormones, they have different target organs and they do different things. You'll see these same two hormones again because they're going to do the opposite in the pancreas. So what gets absorbed in the stomach? A few things. Most everything else gets digested, broken down, moved through. You absorb water, hydrate, electrolytes, sodium, chloride, potassium, such, aspirin, right? And then alcohol. Alcohol gets absorbed in the stomach. So you move on down there and you have an accessory organ, and this is your pancreas. Very important in digestion and such. See there, I say it lies retroperitoneal because it is behind the peritoneum, but it's kind of sandwiched in that mesentery. No sense killing ourselves arguing about that. Two large ducts. There's a pancreatic duct. That's the duct of Versung. And then there's an accessory duct. And this one is superior to it. It's on top of the pancreatic duct. And that's the duct of Santorini. Just sounds wonderful. So here, the pancreatic duct will join with the common bile duct. We'll go over that in a minute, such, from the liver. And that's where the bile from the gallbladder and such is released into the small intestine. And also, the pancreas is composed of these smaller cells, these exocrine cells, and then found throughout, kind of scattered like little bitty islands, if you will, are these islet cells. And they're the islets of Langerhans, and you might call them the pancreatic islets and all that. But when you look at the pancreas slide, I think I put some in that folder for you. I'll have to double check. You'll see them. And once you see them the first time, you'll always see them from there on out. So the pancreas and the spleen can be kind of similarly looking if you're not paying close attention. So you got to pay close attention there. But when you look in the islet cells, there's three types of cells there. There's alpha, beta, and delta cells in that. And I go A, B, D, alpha, beta, delta. And then I go glucagon is with alpha. Insulin was with beta, and the somatostatin is with the delta. What does this pancreas secrete? Of course, it secretes the pancreatic juice. Think of it like that. And you produce about 1 liter to 1.5 liters of that every day. You reabsorb a lot of it. So its main purpose, of course, is to buffer the chyme, stop the pepsin, and then release its own enzymes. You have pancreatic amylase. Remember that amylase goes after starch. Trypsin picks up where the pepsin left off, and it works at it's a enzyme that breaks down proteins at a higher pH, usually 8 to 11. Chymotrypsin, carboxypeptidase, that's helped breaking off carbon groups and such. Elastase goes after elastic tissue. Pancreatic lipase goes after fats. Ribonuclease after RNA, deoxyribonuclease after DNA, and all. So these enzymes are produced and released from the pancreas inactive. If they were to activate within the pancreas, you have pancreatitis, and that's bad. So they have terms for them, and if you look at these names, you'll pick up on this. An intelligent person picks up on this. Trypsinogen, so you take that O-gen off, and trypsin is the active form. Chemotrypsinogen, same thing. Pro is you take the pro off, and you have carboxypeptidase. So proelastase, then take the pro off, you have elastase. You can pick up on that. So here is the mesentery down here. There's your liver. There you can see that sheet of the mesentery, and there, that is the pancreas. This is the duodenum, and these are metal pins that they have folded this up and pinned it out of the way. Okay, so it's kind of lifted up, kind of upside down, if you will. There's a kidney right there. But there's what it looks like. There's what liver looks like, you know. So what regulates the pancreatic secretion? Once again, nerves and hormones. You know, so when you're thinking about stuff, when food gets in your stomach, when you send messages down the vagus nerve and all that, that increases pancreatic secretion. Hormonally, when the chyme from the stomach enters the small intestine releases a secretin, and that kicks up the flow of the pancreatic juice, which has a lot of bicarbonate. And the bicarbonate is what will neutralize the acidic chyme and turn it from an acid to a base. Fats and proteins also then trigger the release of CCK, and that will stimulate the pancreas to release the other enzymes for breaking down fats and proteins. Another organ, the liver, under the diaphragm, covered with peritoneum and such. Two principal lobes, a right and a left, and two smaller lobes, a caudate and a quadrate. Caudal means tail. You'll see that. Quadrate, pardon me, has four sides. The big division between the right and left lobes, as you notice, is this white line on the model, and that's the falciform ligament right there. The liver is composed of lobules and such, and then the lobules are arranged around what's a central vein. There's not capillaries in the livers or sinusoids, which are just passageways and all. And then you have white blood cells found in these sinusoids. These are your Kupffer cells, and they go after bacteria. They help detoxify things, and they remove blood, old worn out blood cells. Your spleen will do some of this, but your liver does some as well. There is a good looking healthy liver. You know, there's that falciform ligament right there. This would be the left lobe. This would be the right lobe. That right there is the vena cava and all. That's probably just connective tissue. That caudate is right. You can see some of it right there. You have to look on the back side to see it. But the caudate is right near that vena cava. And then there's your gallbladder. And that gallbladder is kind of distended. You know, there's it is in centimeters. This is a small person, so that's an inch. 2.54 centimeters is an inch. So you can measure across there. That's about the width of your hand, it looks like, doesn't it? So it's a small person. It's secreted bile. This is a breakdown product and such from the liver breaking down blood and blood products and all. It collects up in these little bitty canals, canaliculi. You've seen that before when you did the bones. And the canaliculi collect up into ductules, and the ductules collect up into ducts, and then you have one draining the right, and you have one draining the left. And then those two ducts come together to make a common hepatic duct. Now, that will keep going and become the common bile. And you go back up, and you'll find a cystic duct. The cystic duct drains the gallbladder. So when the hepatic duct joins the cystic duct, you get the common bile duct. And that then travels out and down through the pancreas, where it meets up with the pancreatic duct. And it then enters into the pancreas at a structure known as the hepatopancreatic ampulla. A little bitty muscle, a little bitty sphincter muscle. They also call that the ampulla of Vater right there. So this is closed. And then when a chyme shows up, this will open. The CCK will cause it to relax and open, and then it'll close. When it closes, the bile will back up and then go back and flow back in through the cystic duct and be stored in the gallbladder. You got to be careful when you do that. If you're dehydrated a lot or you have a really high cholesterol count, that will crystallize and you'll get gallstones, and then you won't be a happy camper. So the liver is fed through the hepatic artery. I had to stop that. The clock was going off. So hepatic artery goes in that. The hepatic portal vein also goes into the liver. And the hepatic portal vein is draining the intestines. So everything you absorb in the intestines travels through the hepatic portal. Stomach and intestines travels through the hepatic portal vein into the liver. A portal vein is a passageway that starts at capillaries and ends in capillaries, but it doesn't pass through the heart. That's a portal system. So a big one in the body is the hepatic portal system because it goes from the small intestines to the liver. So as the blood travels through, you store nutrients, glycogen. You'll turn glucose into glycogen. That's part of the modification. You'll turn glycogen back into glucose, part of the modification and such. Poisons. You pick things up, you detoxify them, they're stored in the liver. And then you'll also produce things. And then you will secrete the glucose back into the body and all. So it will leave through the hepatic vein. Now how it gets to that, you go through the central vein of the lobule, then to the hepatic vein and such, and then into the inferior vena cava. So the hepatic portal vein, hepatic artery, and the bile duct, they will come together at the corners of the lobules, and that's known as the portal triad. And also, you produce about a liter of bile a day. It's yellowish, sometimes greenish and such, and it's mostly water, but it has bile salts. Lecithin is an enzyme that breaks down fats and such. Pigments, you'll have pigments in it. Bile acids, if you will. These are organic acids. Cholesterol is found in there as well. This is what will crystallize and cause the gallstones that then block up the bile duct, the common bile duct. Then you're an unhappy camper. So, bile is a waste, but it's also a secretion. It helps to emulsify, which means takes big fat globules, breaking them down into smaller fat globules that then your body can absorb. That's the purpose of the bile. Bilirubin is a pigment of the bile, gives it its color and such, and that's from the red blood cell breakdown. Newborns, this will overwhelm the liver because the liver hasn't started, and you'll get a jaundice of newborns. People with liver disease, you'll get a jaundicing, which is a yellowing as well. So newborns, it's a physiological. The liver kicks in, starts working, jaundice goes away. And the rest of us, the bilirubin then travels down into the intestines where it's broken down into stercobilin, and that gives the color to the feces. So when you have a rapid excess production of the bilirubin, that's prehepatic jaundice. That's babies. Hepatic jaundice, your liver is messed up. Gallbladder, gallstones, obstructive jaundice, cancer, obstructive jaundice, and such. Boom. So what regulates the secretion of the bile? Nerves and hormones. Nerves and hormones. One secretin right there kicks it up and such. Parasympathetic impulses increase it. Vagus stimulation increases it. Those things will increase the blood flow through the liver. And then when you put a lot of bile salts in the blood, when the body is breaking down a lot of blood, that'll do that as well. And then that'll kick up its release of its materials and all. So, all right, stop that.